Okay, welcome to our next class about carbohydrates. We have completed monosaccharides and today we are going to complete that is called as oligosaccharides. So in my last class I have told about what is the meaning of oligo. Oligo means few. We know oligo means few and the meaning of saccharides means sugar. The meaning of saccharides means sugar. So normally we are dealing with this particular part that where we find that these are sugar or these are carbohydrates having what? Now simple sugar. They are having simple sugar. These are carbohydrates. They are having simple sugars from 2 to 10. Means from disaccharide to decasaccharides. Got it? From disaccharide to decasaccharides, these are the oligosaccharide groups present. So here carbohydrates present that contain simple sugar means they contains monosaccharides. They contains monosaccharides. Got it? So these they do contains monosaccharides and these monosaccharides vary from 2 to 10. Means suppose this is a monosaccharide, suppose this is another monosaccharide, suppose this is another monosaccharide, this is another monosaccharide, this is another monosaccharide, and they are joined by glycosidic bonds, some bond present and the total glucose molecule so formed is called as oligosaccharide. It is called oligosaccharide. Got it? So this is what we call them as oligosaccharides, means few sugar. Few sugar of what? No, few sugars of monosaccharides. And we find these monosaccharides are joined by special bonds present in between them. And these bonds present between them are called as glycosidic bond. Called as glycosidic bond. So these bonds present are called as what? The glycosidic bonds. So these are the glycosidic bonds that combine all monosaccharides to form a complete chain of oligosaccharides. Got it? So this is how we find the 2 to 10 simple sugar are joined by glycosidic bonds to form a chain of molecules that is called as what? The oligosaccharides. Got it? And here also we find same that what? These are polyhydroxy groups of aldehyde and ketones as we have discussed in our that is monosaccharide groups. Got it? Okay, so we have done that in the oligosaccharide, the monosaccharides are joined. The monosaccharides are joined. So there is a joining of simple sugars. These are joined, and these are joined by what kind of bond? That is called as glycosidic bond. Got it? So that they are joined by glycosidic bond, and during the glycosidic bond, there is a release of water. There is a release of water molecules. Got it? So during the formation of this glycosidic bond, there is a great release of water molecules present. Let's see. For example, suppose we are taking a glucose molecule. For example, I am taking 1, 2, 3, 4, 5, 6. Just I am giving an example. Got it? CH2OH. For example, I am taking this. For example, we are taking this type of compound. Suppose for example again I am taking another glucose molecules 1, 2, 3, 4, 5, 6 CH2OH. For example we are taking another glucose molecules. For example I am taking any of the simple sugar. Got it? So normally what happens, to form the glycosidic bond there should be release of water. So in these two molecules will get combined. To combine these two molecules, the main role of this glucose molecules is to have, for example, that is we should have a that is bond should be present. For example, suppose for example, I'm just taking an example. So here what happened, a water molecule is given out. For example, one water molecule is given out. So when we give out the water molecule, so this carbon and this carbon, they are joined by this bond. And this bond so formed are called as what? Now glycosidic bond. Got it? So we call them as glycosidic bond. So here one simple sugar and the second simple sugar. So two simple sugars are joined by one glycosidic bond to release one molecule of water. So one bond produced. Got it? So one glycosidic bond produced and one molecule of water is also produced. One molecule of water is also released. Got it? To form this disaccharide. So this compound so formed is called as disaccharides. Got it? So this is the compound of disaccharides as we came to know. Now it comes what? Now when this compound is formed, suppose the oligosaccharide, we find what we find. Now we find the molecule starts from 2 to 10. So it will release how many water? Now 1, 2, 9 molecules of water will be released. One less. 9 molecules of water will be released. Got it? So in this particular reaction we got what? Now two simple sugars are joined by a glycosidic bond with release of one molecule of water. So we know that oligosaccharides is made up of monosaccharide joined by 2 to 10. So this 2 to 10 oligosaccharide molecules will release 9 molecules of water in total to produce a chain of oligosaccharide. Got it? Okay, so we came to know that suppose we are joining one glucose molecules with other glucose molecules, we'll find what? A disaccharide. We'll find a disaccharides. And this disaccharide contains two simple sugar. And this disaccharide is the form of oligosaccharide. Got it? So disaccharide is now the form of oligosaccharides. But the condition comes when we join a glucose molecules with the fructose molecules. So here what happened? The glycosidic linkage so formed is between 1, 2. So glycosidic linkage formed is between carbon 1 and carbon 2 of the alternative groups. So here we call them as what? Now that is called as 1,2 glycosidic linkage. We call them as what? Now 1, 2 what? Now glycosidic linkage. Got it? So we do call them as 1,2 glycosidic linkage. And this is only present in glucose fructose reaction, and this will produce what? Sucrose. Produce sucrose. And sucrose is what? Is a non-reducing sugar. Got it? Sucrose is a non-reducing sugar. So sucrose is a non-reducing sugar. But in the case of glucose to glucose, when we talk about glucose to glucose, here what we find? Now here the glycosidic linkage is between 1 and 4. Carbon 1 with carbon 4. So it is called as 1,4 glycosidic linkage. Glycosidic linkage. Got it? So this is called as 1,4 glycosidic linkage. But in the case of sucrose, it is of what? 1,2 glycosidic linkage, and it is made up of glucose and fructose molecules. And this disaccharides, whatever we produce, mannose, glucose plus glucose, either you can call them as maltose, whatever we do produce. Suppose this is a maltose we produce, and they are what? They are reducing sugar. These are reducing sugar. These are what? Now these are called as reducing sugar. Got it? So this is what we are giving the formation of bond, chemical bond, or the formation of oligosaccharide with the help of that is monosaccharides. Okay. Now let's move into the nature of disaccharide. Now let's talk about the different types of oligosaccharide. What are the different types of oligosaccharides are found to be present? Okay, let's talk first about disaccharide. Let's talk about disaccharides. So disaccharide means two simple sugar units. Two simple sugar formed oligosaccharide. Oligosaccharides. Got it? So let's go for the example of a disaccharide. For example, we are talking about, let's say we are talking about something about that is called as maltose. Suppose we are talking about maltose. So maltose is a disaccharide, and this disaccharide maltose is normally made up of, I have already discussed isomers and all that thing. So this is normally made up of alpha D glucose. So this is normally made up of what? Now it is normally made up of alpha D glucose plus alpha. So this normally comes in the exam. So it is a dextrorotatory, and these are anomers of alpha. Got it? So maltose is made up of two glucose molecules which are deformed, and they are alpha molecules. Got it? So this is what, and this maltose is commonly called as, commonly called maltose as malt sugar. It is called as malt sugar. So maltose is commonly called as what? Now malt sugar. And this malt sugar is the intermediate compound in starch digestion. It is an intermediate compound. It is an intermediate compound in starch digestion. Got it? So in starch digestion, it is an intermediate compound. So it is an intermediate compound in starch digestion. So as we know, same logic, we apply the same logic as what? Now for example, what we apply the same logic, that this compound so formed is also joined by 1,4 glycosidic linkage. Got it? So they are joined by 1,4 glycosidic linkage because both are of glucose molecules. If there is fructose, then we can call them as, and these are what? Reducing sugar. These are also of what? Reducing sugar. Got it? So these are also reducing sugar, as we all know. Got it? And this mild sugar so present is not so sweet. Mild sugar so present is not so sweet. Normally oligosaccharides are sugar molecules, but oligosaccharides are sugar molecules, no doubt. But this mild sugar, the sweetness of mild sugar is not so high. Got it? Okay, now let's talk to the next disaccharide, that is lactose. That is lactose. The second disaccharide that is present in the syllabus, that is called as what? Lactose. So let's talk about lactose. This lactose is sometimes called as the milk sugar. It is called as what? Milk sugar. This lactose is sometimes called as milk sugar because it is normally present in milk. It is present in both human milk. It is found in both. It is also present in human milk and also present in cow milk. Got it? So this is present in both in human milk and also in cow milk. But in human milk, the maximum percentage of human milk is about 7%, and that of cow milk is 4 to 5 to 6%. So maximum it is present in human milk. Got it? Maximum lactose presence. And normally lactose are chemically made up of, let's see, these are disaccharide, and these are chemically made up of, as we will know, these are made up of beta glucose and beta galactose. Got it? So these are made up of beta D glucose and beta D galactose. So we can say lactose is the combination of glucose plus galactose. So it is a disaccharide. So it's proved that it's a disaccharide. And also they are joined by 1,4, that is glycosidic linkage. It is also joined by 1,4 glycosidic linkage. And also it is a what? Disaccharide of what sugar? Of reducing sugar. It is a disaccharide of reducing sugar. Got it? So here we came to know that we find milk sugar where it is also a disaccharide and also it is a reducing sugar. And let's move on to the last disaccharides that is present in the syllabus, that is called as sucrose. Now let's talk about sucrose. Sucrose is a disaccharide which is made up of glucose plus fructose. Got it? And here we find that both are alpha D. Both are alpha D glucose, both are alpha D fructose. Got it? And here the linkage as one ketone, this is a ketone and aldehyde. So at aldehyde ketones both are combined. So here we find the linkage as 1,2 glycosidic linkage. Glycosidic linkage. Got it? And the sucrose is otherwise called as invert sugar. It is called as invert sugar. And also we came to know that in plants the starch molecules after photosynthesis are transported to different parts in the form of that is sucrose. So in the form of sucrose, the plants transport the starch material. So whatever the starch materials is being transferred is normally in the form of sucrose. Got it? So let's see. This sucrose is a non-reducing sugar. It is a non-reducing sugar. Got it? And normally the sucrose is the sweetest. It is sweet in taste. Got it? It is very sweet in taste. So normally sucrose are what? Sweet in taste. And it is mostly present, or we do call them as what? That is, we call them as cane sugar. Sugarcane sugar. We call them as cane sugar. We call them as, that is called as table sugar. It is sometimes known as table sugar, or sometimes we call them as commercial sugar, as we are taking. Commercial sugar as we are taking. Got it? So you can call them cane sugar, table sugar, commercial sugar, we do call them as. And this sucrose normally produce both compound after hydrolysis, that is glucose and fructose. And we know very well the fructose, they are sweetest monosaccharides. This is the sweetest of all monosaccharides. So sucrose is a disaccharide, but the question comes that it is a non-reducing sugar. It is a non-reducing, is very important. It is known as disaccharide, and the glycosidic bond so formed is between 1 2, carbon 1 and 2. And plants transport the food material starch after photosynthesis in the form of sucrose. Got it? Okay. Okay, next move to the next compound, that is called as trehalose. So trehalose is a type of oligosaccharide or disaccharide normally found to be present. It is normally present in the hemolymph of insects. These are normally present in the fluid, the hemolymph of insects. And these trehalose also contains glycosidic bond. They also contains glycosidic bonds. Yes, they are also found to be present, have glycosidic bonds. And it is a linkage between two anomeric. It is a very important part, two anomeric compound. It is a linkage between two anomeric compounds. Two types of anomeric compounds present, not single type. What? One is alpha type, another one is beta type. Got it? And the alpha, what? Alpha glucose plus beta galactose. So it is a combination of alpha glucose and beta glucose. This is not galactose, this is beta glucose. Got it? Sorry, this is beta. So two anomeric, both of a glucose compound, but galactose is an epimeric, but both glucose compound, but alpha and beta joint are present. So this is present in the hemolymph of insects, and these trehalose, they provide energy to the insects. They provide energy. They provide the energy. So normally these compounds found to be present in the hemolymph provides ample amount of energy to this particular insects. Take the example of cockroach. Got it? Okay. Okay, now move on to the next type of oligosaccharides. That is, for example, you take that is called as trisaccharides. Trisaccharides. So take the example of trisaccharides. So these trisaccharides, for the example, we otherwise call them as raffinose. So these trisaccharides are sometimes called as raffinose, and these raffinose are normally made up of, these are made up of what? That is galactose, glucose and fructose. Galactose, glucose and fructose. Got it? So this is the type of trisaccharide. The name example of trisaccharide is called raffinose. They are made up of galactose, glucose and fructose. So this is one type of trisaccharide. Let's move into another one, that is called as the tetrasaccharide. Tetrasaccharides. So when we move on to tetrasaccharides, these are called as stachyose. STACHYOSE. These are called as stachyose. And the stachyose are made up of four. These are tetra, means there is three saccharides. These are four types of saccharides present. So let's see what are the four types of saccharides we find. So here what we find, for example, this is a combination of, let's see about the combination. So these are the, I think this is the combination of, let's see, glucose plus galactose plus fructose. Got it? So here we find two galactose present, and so in total this is four present. So stachyose is a type of tetrasaccharides. Now let's move the last one, that is called as pentasaccharide. For example, pentasaccharides. So means it must be having a 5, and the best example is called as a barbascose. So the best example is called as barbascose, and normally this barbascose is again made up of, this barbascose present, it is normally made up of, let's see, it is made up of, that is called as galactose, two galactose already present in the above one, and glucose plus glucose, and we find at last fructose. Got it? So 2 galactose, 2 glucose, 1 fructose. So it is made up of 5. Got it? So pentasaccharide, trisaccharide and tetrasaccharide, these are the different examples of your, that is called as oligosaccharides. And it has been found that during the translocation, that is when we are talking about the plant translocation of movement, so the translocation, during translocation in plants, so when there is a movement or transport of plants, this raffinose and stachyose, both these oligosaccharides, they occur in phloem. They are found in the phloem. They occur in the phloem. And the helps, why they are present? Now the helps transport of carbohydrate. The helps transport. So normally, so these two compounds are mostly found to be present inside the phloem, and inside the phloem they are normally present only to help the transport or to help the easy transport of this particular carbohydrate at the time of translocations. Got it? So this is of all about the oligosaccharide, and normally up to this it is asked in your exam. So in a competitive exam, whether it is NEET exam or NTA exam, normally up to this the question are asked. So hope you all enjoyed, and hope you must be having no doubt. If I have any doubt, then please give your comment or give your queries to me in my comment box. And in my next class, I'll give you, we'll discuss about polysaccharide. Okay, all the best. Take care. Thank you.